Physiological trait of aging: genomic instability, telomere attrition, epigenetic alteration, loss of proteostasis and deregulated nutrient sensing, mitochondrial dysfunction, cellular stem cell exhaustion, altered intracellular communication. Physiological trait of aging: genomic instability. Genomic instability: bodies accumulate genetic damage throughout our lifetimes, DNA lesions, nuclear DNA and mitochondrial DNA and nuclear architecture. Exogenous traits like chemical or biological, physical, spontaneous hydrolytic reaction, replication error, reactive oxygen species. Endogenous traits: spontaneous hydrolytic reaction, replication error, reactive oxygen species. Exogenous traits: mitochondrial dysfunction. Mitochondria are especially prone to DNA damage. Mitochondrial free radical theory. Reactive oxygen species cause mitochondrial deterioration, or do they? Evidence suggests perhaps they prolong the lifespan, triggering survival in response to physiological stress. ROS increase in parallel to mitochondrial attempt to maintain survival. Telomere attritions or erosions. The half-life limit is the number of times a human cell can divide. Most mammalian somatic cells don't express telomerase. 50 division is about the limit. Epigenetic alteration. Epigenetic factor can influence how genes are expressed at different ages. Epigenetic alteration. Epigenetic factor change phenotype without changing genotype. Heritable. That change in the chromatin state regulate transcription of genes. Epigenetic alteration. Acetylation of DNA increase the accessibility of the gene, thus increase transcription. The methylation of DNA decrease the accessibility of gene, thus decrease transcription. DNA methylation pattern have been linked to Alzheimer. Chromatin remodeling. Decreasing level of chromatin are associated with aging. Epigenetic chromatin remodeling is caused by environmental stimuli. Loss of proteostasis. As cell age, they lose the ability to do quality control on protein production and are associated with loss of proteostasis, accumulation of damaged and potentially toxic protein. Deregulated nutrient sensing. Multiple nutrient sensing pathways regulate intake, can act to reduce aging of cell because it slows the accumulation of damage due to ROS. But also with age, there is a decline in nutrient sensing, which is turn ages cells somewhat of a paradox. Altered intracellular communication. Neurohormonal signaling become deregulated as inflammatory reaction increase. They deregulate intracellular signaling. Neurohormonal signaling become deregulated as inflammatory reaction increase. They deregulate intracellular signaling, renin-angiotensin system, insulin IGF signaling, adrenergic signaling, immunosurveillance. Cellular sense. The stable arrest of cell cycle and associated phenotypic changes. Cellular senses is intended to prevent propagation of damaged cells and trigger their clearance by immune system. In young age, sporadic damage, cellular sense, tissue homeostasis, block proliferation of damaged cells. There is anti-cancer and pro-aging. In old age, there is damage, repair, clearance, cell renewal. cellular synthesis, block proliferation of damaged cells, anti-cancer, tissue function, inflammation, and stem cell exhaustion, improve aging. Aging associated with an attrition of stem cell exhaustion, anemia, myelodysplasia, osteoporosis, repair of the muscle fiber, intestinal function, reduce in intestinal function, reduce in repair of muscle fiber, increase in osteoporosis and fracture repair, decrease anemia,